was doing the Darth Vader breathing, which I think would be very cool if we all breathed like that. That's why Darth Vader was cool, because he sounded so much cooler than the rest of us. Or everybody else. I mean, like I was in the movie. I wasn't in the movie, but I was Darth Vader. All right, we're in Chapter 9, Campbell's Biology, 7th edition. I know, we're only 7th. We're talking about energy and cellular respiration and all this good fun stuff. So, energy flowing in and out of a system here, coming in through sunlight in the form of plants, prokaryotes, and so on. And it's going to leave as heat. So Chapter 10, we're going to talk about photosynthesis here. And Chapter 9, we're focusing in on the mitochondria and cellular respiration. But they all play a role together in powering or generating ATP. Okay, so first let's talk about some generic background. We've talked about catabolic and anabolic before, but we're going to hit it again. Catabolic pathways produce energy. They break, electrons are transferred, and remember, catabolic pathways break things down. Organic molecules are broken down. It's exergonic. One example of this is fermentation, which we'll learn later, where sugars are partially degraded without the use of or presence of oxygen at a later date. So cellular respiration is actually the most prevalent and efficient catabolic pathway in living systems. It's fairly well conserved. It's found through all eukaryotic cells, and even in certain ways in prokaryotic cells, although we'll get to the details of that at a later date. And one of the important factors for cellular respiration is oxygen, and also the presence of organic molecules like glucose, as well as some others. It could be fats or proteins and things as well, but glucose is the primary one. And most importantly, we get ATP. So respiration, or the breakdown of glucose, is a redox reaction. A what? A redox reaction. That means it involves oxidation and reduction. When something loses electrons, we say that it's being oxidized. When something is gaining electrons, we say that it's being reduced. I know, messed up. When you gain something, you're reducing it. I know, weird. So some redox don't completely exchange electrons, just the amount of sharing that occurs between them within a bond. So this is an example of that. But that's not what we're seeing here. Here's cellular respiration, the overall equation. C6H12O6, which is glucose and 6 oxygens, becomes 6 carbon dioxide, 6 waters, and energy, or ATP here. Now, the sugar itself is becoming oxidized. Okay, it's losing electrons, and those electrons are being taken up by oxygen to make water. And you'll see later that oxygen is actually the final electron acceptor of this entire process. These electrons that are getting shuttled around and moved around like this, exactly like that, get moved onto the oxygen and make water. So here's an example of a redox reaction, and in this case, NAD+, which is a coenzyme, very important as a electron carrier for the electron transport chain, which you'll learn later. NAD+ gains electrons through the help of dehydrogenase, and the NAD+ gets reduced to make NADH, nicotinamide in its reduced form. And you'll see NADH is super important. It's going to take these electrons and pass them off to the electron transport chain. It's going to carry them over there in a way. So it's really important now when we get to the electron transport chain, you'll see, and in general when energy is transformed or passed from one thing to the other, that it's very slow, stepwise reactions. If we do it all at once, we get a very explosive amount of energy that's released, as in the reaction of hydrogen and oxygen to form water. That's an explosive reaction, or the atomic bomb in the same way. So the electron transport chain is a very sequentially slow-moving, or maybe not slow-moving, but a small step passage of electrons. So instead of one explosive reaction, we have very small releases of energy that can be used to form ATP. And here we see that this representation of the electron transport chain. We take the electrons from NADH, we pass them down this series of reactions one by one, and release a little bit of energy to form ATP. And then again, the final electron acceptor, oxygen, makes up water. So we're going to focus in, respiration has 3 main parts. Glycolysis, which is the breakdown of glucose, the lysis of glucose, and it ends up forming 2 molecules of pyruvate. Yes, 2 molecules of pyruvate. You'll see that glucose is a 6-carbon molecule, a 6-carbon sugar, and each pyruvate is a 3-carbon molecule. So amazingly, 3 carbons plus 3 carbons equals 6. The citric acid cycle is the next phase in this, sometimes also called the Krebs cycle, after the guy who discovered it. although he wasn't the only one, but he got his name on it. The other guy got screwed. I don't even know the other guy's name. Citric acid cycle finishes the breakdown of glucose, produces a few other byproducts that will be necessary in the last oxidative phosphorylation step, which is largely made up of the electron transport chain and something called chemiosmosis that we'll see later. This is the main ATP generating step, the most important part. Although ATP is produced in these other two steps, it's very minor. The majority of it comes through oxidative phosphorylation. So here's the big picture. Glycolysis occurs in the cytoplasm. You can see that occurring here. Here's the mitochondria, this yellow bean-shaped structure. Glucose gets broken down into pyruvate. We make a little bit of ATP by substrate level phosphorylation, which we'll explain shortly. You also make some NADH for these electrons that are carried over to oxidative phosphorylation land for the electron transport chain. Citric acid cycle happens in the mitochondria, specifically in the matrix of the mitochondria, and produces a little bit of ATP through substrate level phosphorylation again. Also produces some electron carriers NADH and FADH2, which carry the electrons over to oxidative phosphorylation, the electron transport chain, and uses those electrons, just like the ones over here, to make a whole crap load of ATP through oxidative phosphorylation and chemiosmosis. Now, what was that thing I mentioned? Substrate level phosphorylation. Awkward. Here we have ADP. We want to re-energize it or recharge it. So we take a substrate that has a phosphate group on it. With the aid of one or various enzymes, we move that phosphate group onto ADP to make it ATP again. This is called substrate level phosphorylation. It's the movement of a phosphate group from one molecule to another. So let's talk about an example of this substrate level phosphorylation, that being glycolysis. So glycolysis is splitting of sugar. Again, we break down 6-carbon glucose into 3-carbon pyruvate times 2, and it occurs in the cytoplasm of the cell. Now there's two parts to it. There's the investment, input a little bit of ATP, and then the payoff. You get more ATP out. So here we have the investment phase at the beginning. 2 ATPs are added to help start the breakdown of glucose. And then the payoff begins where we have a series of reactions that make some ATP, again, through substrate level phosphorylation. And in the end, our net gain is 2 ATPs. We also gain 2 pyruvates at the end, 2 waters, and then as we previously mentioned, we also make some NADH, specifically 2 NADHs. That's the electron carrier again. This is the details of the investment phase. You need to memorize all the chemical structures of glucose, glucose 6-phosphate, and so on, and the enzymes all throughout. No, I'm just kidding. You don't need to memorize all that. This is the investment phase. This is the payoff phase. Don't go memorizing all the names of the enzymes and so on, but you do need to know what comes in and what comes out. Okay, next part, citric acid cycle. Checking my timer here. Oh, come on, clock. And I never started my timer. Okay, very good. The citric acid cycle is the next phase. It happens in the mitochondria, of course, specifically the matrix. That's the inner part. So there's membrane, intermembrane space, membrane, and in the middle is the matrix. Okay? Now, the pyruvate that was made in the cytosol, the cytoplasm, from glycolysis needs to get moved into the mitochondrion. Okay? Here we see pyruvate goes through this transport chain, this transport protein, this shuttle, and as it's being brought into the mitochondrion, it has to get converted into a molecule called acetyl-CoA, or acetyl-CoA. This is done by pulling off an electron, so reducing NAD plus into NADH, so we make another NADH here, which is part of the Krebs cycle part that when we count it up. We lose a CO2, as you can see, CO2 gets lost, and then coenzyme A gets added, which you see there. So this is acetyl-CoA. It's a 2-carbon molecule, where pyruvate was a 3-carbon molecule. Okay? Now we're into the mitochondrion, and now we begin the citric acid cycle. Here we lose the NADH, add CoA, lose CO2, and here you have your 2-carbon molecule. Now this is cyclic. Every single pyruvate, remember there were 2 from one molecule of glucose, gets converted into acetyl-CoA, and 1 acetyl-CoA gets added into this cycle. Okay? This 2-carbon molecule will be added to a remaining 4-carbon molecule you'll see a little bit later called oxaloacetate to start this process into a 6-carbon molecule. And then what essentially happens is you have step after step after step of rearranging molecules, phosphorylating different things, creating NADH and so on, making ATP, and then back here with our oxaloacetate, our 4-carbon molecule ready to go again. Okay, so here we see 2 carbon getting added to a 4 carbon, makes a 6 carbon, process goes through, we make NADH, CO2, NADH, ATP. You'll notice an interesting thing, it's actually GTP, so guanine, which gets converted into ATP. Make FADH2, another electron carrier, then more NADH, and then we're finally back to oxaloacetate. We add another acetyl-CoA into the loop. This is in the mitochondria and in the matrix specifically. Okay, and then we take these electron carriers and we move them through to oxidative phosphorylation. This is still within the mitochondrion, but this is now going to happen on the membrane, the inner membrane of the mitochondrion. Okay, during oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthase. NADH and FADH2 will donate their electrons to the electron transport chain, and it's going to power ATP synthesis through oxidative phosphorylation. These electrons that came from glycolysis and from the citric acid cycle in the form of NADH as the carrier and FADH2 as the carrier, these electrons are super, super excited. They're bouncing all over the place. They have energy stored within them. So NAD+, as I like to describe, is the bucket. Okay? The electrons get put into this bucket. Here's one of my crazy electrons. It gets added into this bucket. Okay? But it's so crazy it's going to pop out. So I need to cap this bucket with a lid. Here's my hydrogen ion that I've capped the bucket. Now I have NADH, and now I'm holding this excited electron onto NADH, and I can take this over to the electron transport chain and drop it off. Same with FADH2. The hydrogen ion is the cap holding the excited electron onto the molecule. And so what you notice is that NADH contributes its electrons earlier in the chain than does FADH2. And in fact, for this reason, NADH contributes more energy towards ATP production than does FADH2. You're going to see in just a bit that there's a difference. You can either have oxidative phosphorylation creating 32 ATPs up to 34 ATPs. And that difference is determined by whether it's more NADHs bringing electrons to the ETC or more FADH2s bringing electrons to the ETC, because you get less ATP from FADH2 than you do from NADH. And at the end, as we mentioned, oxygen being the final electron acceptors, the ones that came from NADH and FADH2 gets passed down this chain, slow incremental steps. Finally, the electrons at the end have very little energy available. They get bound to oxygen to make water. And this is the ATP synthase that makes the actual ATP at the end. It looks like one of those baseball caps with the little spinny hat, the little spinny propeller on it, but it's upside down, of course. And imagine these molecules going through the hat, and every time they go through the hat, they make the propeller spin. And as the propeller spins, that energy is added to the anabolic reaction forming ATP. So ADP plus an inorganic phosphate makes ATP. Okay? The power for this, these molecules coming through, they're not actually molecules, they're protons, moving from the intermembrane space where a giant concentration has been built up by the electron transport chain. And as they move through the ATP synthase, it powers this turning of the protein, building and making ATP. Okay. We're going to stop here, and we're going to go to part 2 in just a second because I think you guys need a break. So on this note, enjoy.